Welcome students, welcome to Botany paper explanation 2020 Neet. Question number 91. Which of the following concerning essential elements and their functions in plants? Column 1, A, iron, B, zinc, C, boron, and D, manganese. Column 2, Roman 1, photolysis of water, Roman 2, pollen germination, Roman 3, required for chlorophyll biosynthesis, Roman 4, IAA biosynthesis. If you observe here, iron is essential required for biosynthesis of chlorophyll, and zinc is required for synthesis of auxins, that is indolestic acid biosynthesis. That means indolestic acid is a type of auxin. And boron is useful for pollen germination. And fourth one, D, is manganese. Manganese is a component of OEC, oxygen evolving complex. In presence of OEC, water molecule undergoes breakdown and produces 2 protons, 2 electrons, and half O2. Breakdown of water molecule in presence of light is catalyzed by oxygen evolving complex, and manganese is the component of oxygen evolving complex. That means manganese is required for photolysis of water. The right option for given question number 91 is first option. And question number 93. Question number 93. Which of the following is input into anaerobic sludge digester for further sewage treatment? Option 1, activated sludge. Option 2, primary sludge. Option 3, floating debris. Option 4, effluents of primary treatment. And if you observe in the first option, what is that activated sludge? Activated sludge is nothing but it is substances or microorganisms deposited in settling tank during secondary treatment. That microorganisms deposited contains aerobic bacteria and aerobic fungal mycelium. They are deposited in the form of flocks. These flocks are called as activated sludge. These are transferred into the anaerobic sludge digester for the activity of anaerobic organisms and produces biogas. So that means the right option for question number 93 is first option. And question number 94, the roots that originated from the base of the stem are first option, that is lateral roots. Second option, that is fibrous roots. Third option, primary roots. And fourth option is prop roots. Students, we know that in an embryo there are 2 parts, plumule and radicle. Radicle produces root system, plumule produces shoot system. But in case of monocots, the primary root is short living. After a short period of time, after the degeneration of the primary root, many slender fiber-like roots are developed from the base of the stem. These slender fiber-like roots are called as fibrous roots. These fibrous roots are developed from the base of the stem. Fibrous roots are developed from base of the stem. That means the right option for question number 94 is second option. And question number 95. Question number 95. Floridian starch has a structure similar to first option, laminarin and cellulose. Second option, starch and cellulose. Third option, amylopectin and glycogen. Fourth option, mannitol and algin. And if you observed, first of all, we had discussed about Floridian starch. Floridian starch is the reserve food material present in red algae, that is rhodophyceae members. And this Floridian starch is similar to the structure of amylopectin and glycogen. And the right option for question number 95 is third option. And next question number 96. And here in 96, if you observe, dissolution of the synaptinimal complexes occurs during first option leptotin, second option pachytin, third option zygotin, and fourth option diplotin. And if you observe, in case of leptotin, chromosomes are getting their visibility and movement is initiated. That stage we can call it as bouquet stage. And second option pachytin, pachytin is most significant stage in the life cycle of any sexually reproducing organisms, because during this stage there is a crossing over occurs between non-sister chromatids that leads to the genetic recombinations and ultimately leads to origin of new species. And zygotin is significant because of there is a pairing of homologous chromosomes. The process of pairing of homologous chromosomes is called as synapsis, and each pair of homologous chromosome is called as bivalents. That means bivalents are formed during zygotin. And fourth option diplotin we can discuss, and in diplotin, due to strong repulsive forces between homologous chromosomes, the protein complexes formed during synapsis. What do you call that one? Synaptinimal complexes or dissolution occurs during diplotin stage. That means the right option for given question number 96 is fourth option. And next one is question number 98. That is the process responsible for facilitating loss of water in liquid form from the tips of grass blades at night and in early morning is. So if you observe the question is regarding the process of guttation. What is guttation? Loss of water in the form of water droplets. You know that during transpiration, water is lost in the form of water vapor from the aerial parts of the plant body, particularly through stomata. And during guttation, that is generally we can observe in case of grasses, because in grasses at the tips of the leaves a specialized tissue hydathode is present. That hydathode tissue is useful for guttation. During guttation, water is lost in the form of water droplets. Along with the water, dissolved mineral ions are also lost during guttation. But very important point here, we have discussed what is the driving force for this guttation process. And here they are given options: plasmolysis, transpiration, root pressure, imbibition. So here the driving force for the guttation process, that means loss of water in the form of water droplets, is root pressure. That means for the question number 98, the right option is 3. And next one is we have discussed about question number 99. What is that? The body of the ovule is fused with within the funicle at. So if you observe the structure of the ovule, a orthotropous ovule, once you look at the structure of an orthotropous ovule, and it contains integuments. This is micropyle, and these are the protective coverings, integuments, and this is fertile tissue, nucellus, and this is basal part of the ovary, ovule chalaza, and the stalk of the ovule, that is funicle, and this funicle is attached on the ovule body at a place. That place is called as hilum. So hilum is the place of contact of funicle on ovule body. That means the right option for given question 99 is second option. And next one is question number 100. So if you observe question number 100, how many true breeding pea plant varieties did Mendel selected as pairs? which were similar except in one character with contrasting traits. So if you observed, Mendel selected 14 types of true breeding lines. If the question is regarding how many pairs of or how many traits selected by Mendel, means 7 traits or 7 pairs of contrasting traits. And here the question is regarding how many true breeding lines. So the given options, option number 1, that is 8, option number 2, 4, option number 3, 2, and option number 4, 14. That means Mendel selected 14 true breeding lines. That means the right option for question number 100 is 4th option. And next one is we are discuss about question number 102. The products of reaction catalyzed by nitrogenase in root nodules of legume plants is R. That means they are asking about the biological nitrogen fixation. Once you look at the equation, overall equation of biological nitrogen fixation, that means 1 molecule of nitrogen requires 8 protons, 8 electrons and 16 ATP. In presence of an enzyme nitrogenase, there is a formation of 2 molecules of ammonia plus 2 molecules of hydrogen, 16 ATP plus 16 inorganic phosphates. And they are not asking about energy, and they are asking about only the products. And the products of this biological nitrogen fixation is 2 molecules of ammonia and 2 molecules of hydrogen. This reaction is catalyzed by an enzyme nitrogenase. Nitrogenase enzyme catalyzes this process. That means here the right option for the given question number 102 is first option, ammonia and hydrogen. And next one is question number 103. The number of substrate level phosphorylations in one turn of citric acid cycle. Citric acid cycle, or we can call it as Krebs cycle, or we can call it as TCA cycle, tricarboxylic acid cycle. And in this cycle, only one step, that is conversion of succinyl coenzyme A into succinic acid. And during this step, succinyl coenzyme A into succinic acid, I would like to represent in somewhere else. Here I would like to write, that is, or here I will write succinyl coenzyme A into succinic acid. This is the step where there is a formation of one GTP molecule. That means during this process, the succinic acid and coenzyme A gets separated and energy is liberated. That is useful for fusion of one GDP plus inorganic phosphate. There is a formation of GTP in case of animals, and ultimately it produces ATP. Formation of ATP is called as phosphorylation. Formation of ATP directly due to the involvement of substrate, we can call it as substrate level phosphorylation. Substrate level phosphorylation occurs in case of glycolysis and Krebs cycle. In glycolysis, in 2 steps there is a substrate level phosphorylation. That is, one is conversion of 1,3-DPJ, diphosphoglyceric acid, into 3-phosphoglyceric acid, and another one is phosphoenol pyruvic acid into pyruvic acid. That is the last step of glycolysis. And for substrate level phosphorylation, Peter Mitchell chemiosmotic theory is not applicable. So that means in the step, only one step, there is a substrate level phosphorylation in citric acid cycle. That means the right option for question number 103 is third option. And next one is question number 105. So here the specific palindromic sequence which is recognized by EcoR1. What is the palindromic sequence recognized by EcoR1? Escherichia coli RY13. From that bacteria, this endonuclease enzyme is isolated that recognizes a palindromic sequence. The length of that palindromic sequence is 6 base pairs. And students, you know that what is a palindromic sequence? Palindromic sequence is a sequence of nitrogen bases which reads same on both the strands if the reading polarity is same. Just look at here, GAA-TTC is the palindromic sequence recognized by EcoR1, that is on one strand, and complementary strand also GAA TTC. That means here the reading polarity is 5 prime 3 prime. Here also reading polarity is 5 prime 3 prime. That means the right option for the given question is second option for question number 105. And question number 108. Match the following columns and select the correct option. So column 1, first A, Bt cotton, B, adenosine deaminase deficiency, C, RNAi, and D, PCR. Column 2, first option, that is Roman 1, gene therapy, and Roman 2, that is cellular defense, Roman 3, detection of HIV infection, and Roman 4, Bacillus thuringiensis. Students, if you observe the column A, Bt cotton. Bt cotton is a insect pest resistant variety produced by genetic engineering or recombinant DNA technology, and that is produced by using cryogenes isolated from a bacteria, Bacillus thuringiensis. Those genes are producing crystalline proteins. Those are protoxins which are inactive. When they enters into the insect gut region, they becomes active toxins and protecting the plants from the effect of insects and pests. And second one is we are discussed about adenosine deaminase deficiency. That we can call it as ADA deficiency. ADA deficiency leads to SCID, severe combined immunodeficiency. And that kind of disease is treated by means of two methods. One is bone marrow transplantation, and second one is gene therapy. So what is gene therapy? Replacement of a default gene with functional gene is called as gene therapy. That means the correct combination for B is Roman 1. And RNAs, what is RNAs? RNA silencing or RNA interfering or gene knockdown or quelling. All these are the different names of RNA silencing or RNA interference. So this is common cellular defense method in most of the eukaryotes. That means C is combination, correct combination is Roman 2. And fourth, PCR, polymerase chain reaction. Nowadays, polymerase chain reaction is an advanced technique useful for detecting mutations in suspected cancer patients, as well as it is also useful for early detection of HIV before the appearance of symptoms, and as well as in DNA fingerprinting in forensic science. For DNA fingerprinting process also PCR technique is used. That means the right option for the question number 108 is second option. Next question is 109. What is that? 109. Students, if you are observed, identify the substrates having glycosidic bonds and peptide bonds respectively in their structures. So first option they are given inulin. If you are observed, in case of inulin, inulin is a polymer of fructose. In that case, the fructose molecules are linked by glycosidic bond. And the next one is insulin in the first option. Insulin is a polymer of amino acids. It is a polypeptide chain. That means amino acids are linked by peptide bonds. That means the first option is the right option for question number 109. And next one is we are discuss about question number 112. What is that? Question number 111. Question number 111, if you are observed, name the enzyme facilitates opening of DNA helix during transcription. And if you are observed in this question, and I observed so many students are troubled in this question, they thought that the right answer is DNA helicase. Because students, you observed once the question they are mentioned that facilitating the opening, not opening of DNA. They are asking facilitating opening of DNA. The first option is RNA polymerase, second option is DNA ligase, and third option is DNA helicase, and fourth option is DNA polymerase. And the right option for this question number 111 is RNA polymerase, because RNA polymerase facilitates the opening of DNA helix, and opening of DNA helix is performed by DNA helicases. So that means right option for question number 111 is first option. And question number 112, which of the following is correct about viroids. What are viroids? Viroids are small infectious microorganisms. They are submicroscopic biological entities. And if you observe, in case of viroids, viroids are infectious nucleic acid. That means only nucleic acid is present. Even virus contains nucleic acid and protein. Prions contains only proteins but not nucleic acids. Viroids contains only nucleic acid. What type of nucleic acid it contains? That is SS, single-stranded RNA. Single-stranded RNA is acting as the infectious agent, causes diseases in plants. Best example for the diseases caused by viroids is potato spindle tuber disease caused by potato spindle tuber viroid. This is an infectious nucleic acid RNA without protein coat. That means the right option for given question 112 is third option. They have free RNA without protein coat. And next one is we are discussing about question number 113. Match the following. Column 1, A, inhibitory activity of inhibitor of catalytic activity, and second one is possess peptide bonds. Peptide bonds means regarding they are asking about the proteins. And C, cell wall material in fungi. And fourth one is, that is D, is secondary metabolite. And in column 2, Roman 1 ricin, Roman 2 melanate, Roman 3 chitin, Roman 4 collagen. And if you observe in this, the right combination A, that is inhibitory activity, catalytic activity means the enzymatic activity is inhibited by. In the given column 2, we can observe melanate. Melanate is a competitive inhibitor to the enzyme succinic dehydrogenase. It is competing with the succinic acid, inhibiting the activity of succinic dehydrogenase. And next one is we are discuss about possess peptide bonds. Peptide bonds are present in proteins. In the given column, collagen. Collagen is the most abundant animal protein. It is an intracellular ground substance present in the animals. That is a polymer of amino acids. Those amino acids are linked by peptide bonds. And cell wall material in fungi. In fungi, cell wall is composed with chitin. And D, secondary metabolite. Ricin is a secondary metabolite. The right option for given question number 113 is second option. Correct combination is. And next one is students, question number 115. Some dividing cells exit the cell cycle and enters a vegetative inactive stage. This is called as quiescent stage or G0 stage. This process occurs at the end of. Students, this is very interesting question. In the previous year, last year also they are given this kind of question. Last year they are asked about in G0 stage, what is the condition of the cell? How the cell is in G0 stage? In G0 stage, the cell is in cell cycle is suspended. That means cell division process is suspended and cell is active in metabolism. And here they are asking now how the cell is enters into the G0 stage. That means by exit out from Z1, exit after completion of Z1, it enters into the G0. The right option for given question number 115 is third option. And next one is we are going to discussing about question number 116. Match the organisms with its use in biotechnology. First option in column A is Bacillus thuringiensis, B is Thermus aquaticus, C is Agrobacterium tumefaciens, and D is Salmonella typhimurium. And in column 2 they are given Roman 1 cloning vector, Roman 2 construction of first rDNA molecule, Roman 3 is DNA polymerase, Roman 4 is cryproteins. If you observe Bacillus thuringiensis, in previous question only I mentioned about these cry genes. Insect specific genes are isolated from a bacteria Bacillus thuringiensis, which are used in the production of insect and pest resistant BT cotton variety. So on the same topic they are given two questions. And here Thermus aquaticus. Thermus aquaticus is the source of heat resistant DNA polymerase, that is Taq polymerase, that is extracted from a heat resistant thermophilic bacteria, that is Thermus aquaticus. It is a DNA polymerase enzyme that is used in PCR technique. So that is B is correct matching with Roman 3. And C, agrobacterial tumefacients. We know that to produce transgenic plants, the most common using vector is TI plasmid of Agrobacterium tumefaciens. TI plasmid of Agrobacterium tumefaciens, and that is the most common using vector in the production of transgenic plants. What is the most common using vector in the production of transgenic animals is that is adenovirus. Adenoviruses are most common using are retroviruses, and here TI plasmid of Agrobacterium tumefaciens is most common using vector, cloning vector. And D is that is Salmonella typhimurium. So the Salmonella typhimurium is used in the construction of first rDNA. First rDNA is developed by Stanley Cohen and Herbert Bayer. They are developed disease-resistant E. coli bacteria, not disease-resistant, antibiotic-resistant E. coli bacteria. They are isolated antibiotic-resistant genes from Salmonella typhimurium. That is why the right combination for given question number 116 is third option. And next one is question number 118. This is also same question. Just now only we have discussed cell exit out from G1 and enters into the G0, that is quiescent stage. And here also similar identical question, that is 118. Identify the correct statements with regard G1 phase, gap 1 phase of interphase. And here first option, nuclear division takes place. So nuclear division means karyokinesis. And second one is DNA synthesis or replication takes place. DNA synthesis or replication takes place during a S subphase of interphase. And third one is recognition, reorganization of all cell components takes place. And fourth one is cell is metabolically active, grow but do not replicate its DNA. So the fourth option is right option. During G1 phase, the cell is metabolically active and there is a synthesis of RNA and proteins, but DNA replication, centriole duplication does not occur. That means the right option for question number 118, that is 118, is fourth option. And next one is question number 119. The first phase of translation. This is a very important question. It is not regarding the initiation. Students, you must be very careful. It is not regarding initiation of polypeptide synthesis. It is first phase of translation they are asking. And the first option is recognition of an anticodon. Second option is binding of mRNA to ribosomes. Third option is recognition of recognition of DNA molecule, and fourth option is amino acylation of tRNA, that is a charging of tRNA. The first phase of translation is amino acylation of tRNA. That means activated amino acid is attached to the 3' end of tRNA. That itself is called as amino acylation of tRNA or charging of tRNA or activation of tRNA. The right option for the given question, we can discuss exactly. It is binding of mRNA to ribosomes is initiation of translation. And question number 124. Question number 124. Ray florets have first option, half inferior ovary, superior ovary, hypogynous ovary. So here question number 124, we can observe inferior ovary is present in ray florets, because in case of ray florets belongs to the family Asteraceae. Asteraceae shows head inflorescence. Head inflorescence is present, and in case of head inflorescence, the ray florets, these are unisexual female. Even we can observe in case of marigold. These ray florets, ovary is completely embedded within the thalamus. That means inferior ovary is present. This is the gynoecium, and here ovary is completely, this is ovary, and this one is thalamus. Ovary is completely embedded within the thalamus. That is why it is epigynous flower or having inferior ovary. That means the right option for question number 124 is second option.